Hi there, this is lecture 2 of the musculoskeletal system, and specifically we're going to be talking about the muscular system. There's over 600 muscles in the body as opposed to only about 206 in the skeletal system. Muscles make up 40 to 50% of the body weight and have many functions. They permit movement, produce heat, stabilize joints, and maintain posture. All of this depends on a unique feature of muscle tissue, and that's excitability, which is the ability to respond to signals from the nervous and endocrine signals. This diagram here shows the basic structure of a muscle. You'll note that a muscle cell is here in the center, and I'm circling that. A fascicle is made up of dozens of muscle cells or muscle fibers, whichever you want to call it. You can see that the blood vessels and nerves dive deep into the muscle and come in to innervate the different muscle fibers, but first it has to penetrate the fascicle to get down to the muscle cells. Around the fascicles, you have the paramycium here, and around the entire muscle, surrounding everything, is the epimycium. And then around each muscle fiber or cell, surrounding all of those, is the endomycium, which is a lot of that is fat tissue to provide energy for muscle contractions. The fascia is the dense connective tissue covering around the entire muscle. It's really a continuation of the epimycium, paramycium, and endomycium. All of those combine with the fascia to form tendons and aponeuroses here, which of course attach the muscle to the bone. Within the muscle fiber itself, at the microscopic level, you have the sarcolemma, which is the same thing as the plasma membrane. Sarcoplasm is the same thing as cytoplasm. So if you haven't guessed by now, sarco means muscle. Sarcoplasmic reticulum is the same thing as the endoplasmic reticulum, and this is a very important part of the muscle cell. This is where calcium is stored, and it's released from the sarcoplasmic reticulum when it's stimulated by a nerve impulse. The sarcoplasmic reticulum has a complex intracellular network of tubules and sacs, and the T-tubules are extensions of the sarcoplasm that go in and connect with the sarcoplasmic reticulum. The sarcomere is the basic contractile unit, and so a muscle cell or muscle fiber is made up of thousands and thousands of these sarcomeres connected end to end. And then each one of these little units contracts, and that brings the entire muscle together. So a sarcomere is bundles of myofibrils, and they basically delineated by Z-discs. So 2 Z-discs make up a sarcomere. Everything in between the Z-disc is a sarcomere. Again, T-tubules are the inward extensions of the sarcolemma. These are similar to cardiac gap junctions that allow the penetration of electrical signals deep into the muscle fiber, and these are closely related to the sarcoplasmic reticulum, as I said earlier. And then myofibrils are composed of myofilament bundles, and myofilaments are myosin and actin. Myosin is the thick protein strand, and actin is the thin protein strand. It's important to remember that myosin stays stationary and actin slides back and forth over myosin. So this shows the basic structure of the sarcomere. You have right here is the Z disc, and then here is another Z disc here. So everything in between this is a sarcomere. And then you have the thick filament in blue, thin filaments in red. Thick is myosin, thin is actin. This is a muscle fiber or a muscle cell. Again, you have the sarcolemma and the sarcoplasm. You have the nucleus, and here again is one sarcomere with the Z disc at each end. You can see that the T-tubules are extensions of the sarcolemma deep into the muscle cell. It's full of mitochondria scattered intimately in between all of the myofibrils. And in yellow you have the sarcoplasmic reticulum. And this is where calcium ions are stored for muscle contraction. So the physiology of a muscle contraction is basically the sliding filament theory. And what that says is that the myosin and actin combine, and there's a dynamic process of actin sliding over myosin to cause the entire muscle to contract. It says that fibers are at rest until stimulated to contract, and the stimulation requires a neuromuscular junction, and that means that there's a site where the muscle is connected to a nerve fiber. That is called the motor end plate here. So what happens is small vesicles in the cytoplasm at the end of the nerve secrete acetylcholine or acetylcholine. That diffuses very quickly across a little cleft in between the nerve and the muscle, and it's a physical gap. It's only like 10 to the minus ninth meters in size, which is tiny, but it is a physical gap. When the acetylcholine connects with the receptor, that stimulates the sodium ion channels, and sodium rushes into the cells, which causes depolarization. The depolarization dives down into the T-tubules because that's an extension of the plasma membrane or the sarcolemma, and that works on the sarcoplasmic reticulum to secrete calcium ions. We'll go through action potentials and nerve impulses in detail in the next chapter of the nervous system, but for now, just remember that it's membrane depolarization, and calcium is critical in the muscle cells. So here is, again, this is the motor end plate, is the structure on the sarcolemma that's directly adjacent to the synaptic vesicles of the nerve. This entire structure here is called a neuromuscular junction. So you can see here is the motor neuron. The impulse comes down here to the end, where it stimulates the release of acetylcholine in the synaptic cleft, which is here. That binds to the receptors, the acetylcholine receptors on the motor end plate, which those are shown here. That in turn stimulates an action potential, which then moves down the muscle fiber, but it also dives deep down through a T-tubule here, where it connects with the endoplasmic reticulum shown over here. So if the impulse comes down here, it's then going to work here on the endoplasmic reticulum, which then secretes calcium ions. And the calcium ions then work on the myosin heads, to allow muscle contraction, and we're going to show that here in this slide. So here we have actin and myosin, and you see these little golf club sort of ends on the myosin is the myosin heads, and this is the sliding filament theory. So what happens in step one, we also have tropomyosin, and this, what these are doing is blocking the actin from connecting to the myosin. And then troponin here is the receptor for the calcium. So what happens first is ATP binds to the myosin, the relaxed myosin head, and calcium comes in and connects. What's important to remember is that the ATP connects to the myosin head and allows that to move into a resting position. So this is the resting position of the myosin head. Calcium comes in and connects to troponin. Troponin then rotates and moves the tropomyosin into a position where the actin is exposed. This allows the actin and myosin to bind here, that's shown there, and that displaces the ATP, which is now cleaved to form ADP and phosphate, and that releases the energy. The release of the ADP from the myosin head allows the myosin arm to contract, and that pulls the actin this direction. And then once the ATP comes and binds to this head, it allows it then to go back here and go into the resting position, and the entire process starts again. When one muscle fiber is stimulated to contract one time, that's called a twitch, a muscle twitch. But tetanus is when there's a sustained contraction. It's not tetanus like the disease tetanus, but tetanus refers to the sustained contraction of a muscle that occurs when we want to have any significant movement of a body part or our body. This shows what happens at the sarcomere level, and each sarcomere has several different bands and discs involved. Basically, here's the Z discs again. This is what you always look for first. And then the I band shown here is essentially myosin. The H zone here is the hot zone, and that's where the action is occurring between actin and myosin. So you can see that the A band includes, basically, it's where the actin is, and it includes the hot zone, or the H zone. Okay, so here's the A band, the I band, which is only actin, and then the H zone initially is only myosin. So you can see what happens is the Z disc or the Z lines remain the same size. They don't shrink. What shrinks is the I band, and you can see that here. Okay, you can see that the A band remains the same size. Okay, it doesn't change in size. However, the H zone changes because the actin moves into this space here. Okay, so the A band and the Z disc remain constant in size. Here, at the end of a contraction, the A band is essentially the same length as a Z disc. Okay, and the I band is what shrinks. That's actually what disappears during a muscle contraction. This is an actual electron micrograph showing a muscle fiber, and you can see these different structures. The sarcomere is shown here. You have the A band right here, which is actin. You have the H zone here in the middle. And then you have the I band right here, which is just myosin. Okay, so when this contracts, again, the A band is going to stay the same. The Z disc is going to move in here and in here. And the H zone will change color, so it's all completely dark. This H zone will disappear and become the same color as this and this. And that's what happens when the muscle contracts. This is another way of looking at it. If each one of these little guys here, they represent a myosin head. And so as they pull and then move forward on the rope here, which is the actin, they pull it again, and you can see that they're pulling from each side. So this is a nice example of what's going on. There's two different types of what we call metabolism or exercise. It's anaerobic and aerobic. And you've all heard of this. Anaerobic refers to a state where oxygen is not abundant enough to drive metabolism, whereas aerobic activity means that there's plenty of oxygen to drive metabolism. The difference is a significant amount of ATP. There's about 18 times more ATP produced during aerobic activity as opposed to anaerobic. However, aerobic requires time to kick in, usually up to about a minute before it starts to resupply the cell with ATP. So during that initial phase, the cell has to rely on anaerobic processes to move. So anaerobic relies on ATP stores that are just in the cell, and it also relies on creatine phosphate, which quickly changes ADP back into ATP so it can be used again. Glycolysis occurs, which is the rapid breakdown of glucose in the cytoplasm. So what happens in muscles that are dialed in and tuned for anaerobic activity, the muscle bulks up significantly, and it bulks up because the muscle wants to be as crammed full of ATP, glucose, and creatine phosphate as possible, and so that during activity there's an immediate availability of these substances. Aerobic activity, on the other hand, the muscle cell wants to be packed full of mitochondria and maintain a relatively slim configuration to optimize weight to power ratio. It relies again on oxidative phosphorylation, which is the Krebs cycle and the electron transport chain that we talked about in the first unit. Increased numbers of mitochondria and optimized oxygen delivery, which means the cardiovascular system has to be functioning really well. So a motor unit is the motor neurons plus the muscle fibers that they innervate, and there's a ratio of motor neurons to muscle fibers. Okay, so remember that ratio, neurons to fibers. If you increase that ratio, which means that you increase the number of neurons and or decrease the number of muscle fibers innervated by a neuron, that indicates fine coordination of muscle movement. For instance, in our hands, the motor unit ratio will be much higher than it is in our thigh muscles, for instance, because our hands are tuned for very fine movement. Our tongue and mouth have a much higher ratio than our shoulders. And this diagram down here shows that within the spinal cord, we actually have different motor units. We have a motor unit 1, 2, and 3, which is shown here. The motor unit 1 is pink, motor unit 2 is red, and motor unit 3 is the orange here. And you can see how each part of the nervous system here innervates different types of motor units, and that's where we get fast and slow twitch muscles. And so we're born with a certain ratio of fast and slow twitch, and then we can also develop that through exercise and activity. There's an optimized preload for muscle contraction, and that's kind of obvious. So if the muscle's stretched out too far, you can see that there's no overlap initially of the actin myosin, and so the sarcomere is stretched out so far that it requires a lot of work to start to contract that muscle. Similarly, if the muscle is crammed in so tight together, there again is nowhere for the actin to go. So there's an ideal preload for muscle for tetanus type contraction or sustained contraction. This becomes extremely important in the cardiovascular system, and we'll talk more about that when we get there. But the heart has to have a certain amount of blood in the chamber that stretches out the sarcomere a little bit so that it can contract most forcefully. If the heart's too stretched out, like here, like in congestive heart failure, it can't beat very well. Or if it's crammed together as here, it also can't beat very effectively. So this is just illustrating that there's an optimized part in the sarcomere called preload for that muscle to contract. And we have isotonic and isometric exercises. These are methods that we can use to strengthen muscle. Of course, we all know about weight training and just regular exercise, which is isotonic. That means same tension but a changing length. So like if you're lifting something, that's an isotonic movement. And isometric has become more popular lately with yoga and other activities, and we're finding that it's equally very effective for building muscle tone and maintaining muscle health. And here the muscle, the length of the muscle stays the same, but the tension changes. Okay, so you put the muscle into a configuration, an optimal configuration, and flex that muscle over time. This shows the configuration of the cardiac muscle. You'll note that it does not have tapered ends. It does have intercalated discs, and these are very important. They're gap junctions in between the cells. You can see those here and here that connect the different muscle cells together so that a nerve impulse coming down will spread not only in the cell itself very well, but it'll go and into this cell and into these neighboring cells very quickly to cause those to contract, and that increases the efficiency dramatically. Cardiac muscle has a very unique quality called self-excitation, and that means that the sinoatrial and atrioventricular nodes do not require input from the central nervous system to function, and they discharge at a continuous rhythm. This is why when you take a heart out for a heart transplant, you put it on ice, because the heart will continue to contract at a regular rhythm. Now, the heart muscles are very finely tuned with nerves from the central nervous system and also hormones that can cause the heart to beat slower or faster with more force or less force. Smooth muscle has these spindle-shaped fibers with these tapered ends like this. They're much smaller than skeletal or cardiac muscle cells. There's only one nucleus per cell as opposed to cardiac muscle and skeletal muscle that can have more than one nucleus per cell. Smooth muscle has very slow, sustained contractions. It doesn't have the quick, powerful stroke of a skeletal muscle or cardiac muscle. Like the heart, however, it's under non-voluntary control. However, it requires connections with the central nervous system to work. It lacks sarcomeres, but it does contain actin and myosin in sort of a loose affiliation. So there's not this regular structure that you see with cardiac muscle and skeletal muscle. We see these in the intraocular muscles in the eye. We also see it around glands and around arteries and veins is the primary locations of smooth muscle. Also, the wall of the gastrointestinal tract and the walls of the airways is where we find smooth muscle. Skeletal muscle has many different configurations. There's parallel muscles, convergent muscles, circular muscles, fusiform, spiral, and pinnate muscles. And these are all examples of these different types. It's important to remember that the origin, every muscle has an origin and an insertion. The origin is, you can think of that as the beginning of the muscle. The insertion is where it inserts onto the bone. Remember that the origin never moves during muscle contraction. It's only the insertion that moves relative to the fulcrum. Every muscle has the prime mover, also called the agonist, and every agonist has an antagonist. There's usually several agonists. There's one agonist, but then every agonist has several synergists that work with the agonist to produce a movement. So the prime mover, again, is called the agonist, and it's associated with one or more synergists, as shown here. Antagonists oppose the action of the agonist. This shows the lever system here quite nicely. So E is the force of contraction here. R equals the load, and then F is the fulcrum. So a first class lever has the fulcrum in between the load and the force. Second class lever has the fulcrum with the load in between the fulcrum and the force. And then a third class lever has the load at the end with the fulcrum and the force shown here. So the force lies in between the fulcrum and the load. So the elbow joint that we looked at earlier is an example of a third class lever. The ankle joint here is a second class lever. And you can see that movement around the axis, or the first, the axis and the atlas, C1 and C2, is a first class lever. You can see that third class levers are not very efficient, and that's because the load lies outside of the force. However, if the load, as the load moves in closer to the fulcrum, it becomes much easier to lift this lever relative to the fulcrum. And if you think about that, it's true like with a wheelbarrow. A wheelbarrow is an example of a second-class lever, where R is the load and the E is the force lifting it. And if you think about a wheelbarrow, if you extend the lever, say, out to here, thereby effectively moving the load closer to the fulcrum, it becomes much easier. Then it requires less force to lift the load. Okay, the rest of the lecture basically is just going to focus on muscle groups. It involves a lot of memorization, and there's not a lot of explaining to do. The facial muscles shown here, the major groups, is the occipital frontalis, which is the occipital and frontal muscles. They're connected by an aponeurosis, which is here. This is the aponeurosis. And then this is the frontal muscle. And then in the back you have the occipital muscle back behind the head. The corrugator supercilii are shown here, but this whole area right here is called the glabellar region. And you have the procerus muscle, which is a large muscle right here in the center, and then you have the corrugator muscles here. These are the major muscles, and then the corrugator is underneath the frontal muscle here. These are the muscles involved in upper facial expression, like your eyebrows. It's also the area where irides or wrinkles form, which is why this the upper face here is the area that's most responsive to Botox injections. And so Botox in these areas is fabulous for removing wrinkles and making people feel younger. Orbicularis oculi encircles both eyes, you can see there. The orbicularis oris and buccinator muscle shown here. The buccinator muscle is deep here. The buccinator allows us to hold our food against our molars when we're chewing. It also allows us to move the lips in and out and to kiss and do other things like that. The zygomaticus major is shown here, major and minor, and this is what pulls the mouth up when we smile. The zygomaticus major and minor muscles. These other muscles are there just for reference, but these are the ones that I've listed are the ones that you will be responsible for on the exam. These are the ones that you want to become very familiar with. Muscles of mastication include the masseter muscle here. This is the major muscle right there. The temporalis, which is up in this region, and also the buccinator muscle here. And these three muscles, this is with the masseter muscle cut away. And this one also, you can see that this is looking inside the mandible, so you've got the mandible is cut away here to show the pterygoid muscles. The lateral and medial pterygoids allow us to move the jaw this way, laterally. The masseter, buccinator, and temporalis allow us to chew up and down like this. And this shows the insertion of these different muscles on the mandible. Neck muscles, there's a lot of neck muscles, but the ones you should know about are the splenius capitis, pulls the chin up. The sternocleidomastoid muscle here, also called the prayer muscle, pulls the head down. However, it also moves the head side to side. What's interesting about the sternocleidomastoid muscle and the trapezius muscle is that when they contract, they move the head to the opposite side of contraction. So if I contract the right sternocleidomastoid and trapezius muscle, but primarily the sternocleidomastoid, it will actually pull my head to the opposite side and vice versa. So these are the major neck muscles that move the head, and the insertion of the sternocleidomastoid is on the mastoid bone. Here, the mastoid process of the skull, and then the clavicle. Muscles of respiration are actually quite complex. We think of the diaphragm as being the primary muscle of respiration, and that's true for regular respirations. It's primarily the diaphragm, but when we do forced respiration, such as inhaling forcefully or exhaling forcefully, then we're using more of the thoracic muscles. And so the muscles of inspiration are here: sternocleidomastoid, scalene, the external intercostal muscles in the diaphragm, and are involved in forced inspiration. And then during forced expiration, you have the internal intercostals. Also, the diaphragm is involved, and then the abdominal muscles as well down here. So remember that the origin and insertion of the intercostals depends on whether they are external or internal. They look very similar, but they have a different origin and insertion. Abdominal muscles shown here. You have the rectus abdominis that's covered by the rectus sheath. You have the transverse abdominis, and then the external obliques here are the primary muscles of the abdomen. Muscles of the back, there's many muscles of the back, which is why people frequently have back pain, is because it's very complex. But the major ones are the trapezius, which again move the head laterally and also extend the head. The splenius capitis also extends the head. The semispinalis capitis does the same thing. The rhomboids pull the shoulder, or adduct the shoulder blade. We'll talk about the supraspinatus, infraspinatus, and teres minor when we talk about the shoulder. The teres major is responsible for pulling the arm back, or adducting the arm. You have the latissimus dorsi, called the lats, are a major muscle of the back. This is connected to the lumbar and sacral spine by fascia called the thoracolumbar fascia. The erector spinae muscles are long longitudinal muscles in the deep back region. So really you have the erector spinae muscles, all of these together, combined with the trapezius, the lats, and then in the middle you have the splenius capitis and the semispinalis muscles. And really that's it. So deepest are the erector spinae, middle layer semispinalis and splenius, and superficial trapezius, the lats or latissimus dorsi, and that's about it for the back muscles. The pelvic muscles are pretty easy because the levator ani and coccygeus muscles here make up the pelvic diaphragm, which is 90% of the entire pelvic floor. So these two muscles basically make up almost everything. And then the piriformis is just this small, relatively smaller muscle back here that makes up the rest. Of course, this is the anterior, so here's the symphysis pubis. In the female, you have the urogenital diaphragm here. You have the vaginal orifice here, and then you have the anal canal here. So these are the different muscles of the floor of the pelvis. This is, the previous slide was actually, if you were standing looking down into the pelvis. This is as a person is looking externally. So when, during a pelvic exam or during childbirth, this is essentially what is visible, and you have the superficial and deep shown here. Again, you can see the urogenital diaphragm is here, and that's made up again of the levator, ani, and coccygeus. These form this sort of kind of a hammock across the pelvic floor. The superficial transverse perineal muscle shown here is the most superficial muscle. Here's the piriformis again that we talked about. So when an episiotomy occurs, that's a tear that occurs in the vaginal orifice that extends in this region here, and it's usually, you can do one manually with scissors, or it can just tear during childbirth. But muscles of the appendicular skeleton mean muscles associated with the shoulder girdle, the pelvic girdle, and the limbs of the body. The shoulder girdle muscles basically involve the shoulders, the rotator cuff shoulders, and then the major job is to keep the scapula down against the thorax, help with forced inspiration during breathing, and to also allow us to raise the shoulders and to rotate the shoulders. So the rotator cuff muscles are actually pretty easy to remember. You have the supraspinatus, subscapularis, infraspinatus, and then the teres minor here. Superficially, the trapezius and deltoid, and then deep to that, you have the rotator cuff muscles and the rhomboids, which are responsible for adducting the scapula or pulling them inward like this. These are the rhomboids right here in this region. This just highlights the rotator cuff muscles and shows exactly what they are. So you have the supraspinatus, the infraspinatus is this muscle complex here, and then the teres minor here, and then if you're looking at this is a posterior view. If you're looking anteriorly, then you have the subscapularis, which is this large triangular muscle right here, and these the subscapularis forms the anterior part of the rotator cuff, and these three muscles form the posterior aspect of the rotator cuff. The muscle joint has the greatest range of motion, but it also is the most susceptible to injury. The muscles of the upper limb are shown here. The coracobrachialis, shown here. This is an anterior view, so you have the coracobrachialis that goes from the coracoid process of the scapula down to the inner aspect of the humerus. And then anteriorlis goes from the humerus down to the ulna, and is shown here. And these lie deep to the biceps brachii. So anterior muscles are here and here. Posterior muscle is here. And you can see those here. The pronator quadratus is at the wrist level. The pronator teres and the supinator are the three that you should know. You can also see here, this is called the retinaculum on the dorsal aspect of the wrist right here. The extensor indicus goes to the index finger. We've already talked about the supinator. So these are really the muscles that the extensors you should be aware of are just this major group here. The extensors, which include all of these. The flexors are shown here going superficial to deep. These are the flexor carpi radialis, the palmaris longus. Here's the flexor carpi radialis, the palmaris longus, and the flexor carpi ulnaris here. In the middle layer, we have the flexor digitorum superficialis and the flexor pollicus longus are the only two. And then deepest is the flexor digitorum profundus, and that profundus indicates deep. And we've already talked about the flexor pollicus longus. So here, the flexors again, to recap, going from deep is the flexor digitorum profundus, flexor digitorum superficialis, flexor pollicus longus. Most superficial is the flexor carpi radialis, the palmaris longus, and the flexor carpi ulnaris. And that's it for the flexors. Muscles of the hand, I don't expect you to know really any of these except for the opponent's pollicus is very important, as well as the adductor pollicus. The reason that these are important, the opponent's pollicus, actually these three, is because it allows us to apposition the thumb with the digits, and that is a unique feature of human beings, for an enormous amount of dexterity with digits to perform very highly coordinated and very complex maneuvers with the hands. And so really it's these three muscles that are important. The gluteus maximus, of course, and the gluteus medius right underneath that. And then the iliotibial tract is important. It's a dense connective tissue tract, and it's frequently used to harvest tissue for many different types of surgeries. The hamstring group is used for flexing the lower leg, and we have those shown here. The biceps femoris, semitendinosus, and semimembranosus. The vastus medialis, the rectus femoris is the large middle muscle, and then the vastus lateralis. These are the extensors of the lower leg. The thigh flexors are primarily the iliopsoas group. The thigh adductors used for pulling the thigh inward is essentially the gracilis, the pectineus, and these three muscles here. Muscles of the lower leg, posterior, the flexor digitorum longus, and then the popliteus and flexor hallucis longus here, and that's really it. Posteriorly, you have the gastrocnemius, which is the calf muscle. You have the lateral and medial heads. The soleus muscle works together with the gastrocnemius. The deep muscles of the lower leg is the tibialis posterior here, the flexor digitorum longus, the flexor hallucis longus, and that's it. I do not expect you to know the muscles of the foot. These are here for reference only.